Welcome to our second plenary session this morning. This session features some selected poster presentations that we thought we'd bring into an oral format this year. We've got a slide, should be a slide up in the background there, with summary presentations. I'm going to call upon the first of these people now to come and chat. This will be Jenny Tran. Jenny's a PhD grad student at Vancouver Coastal Health. And Jenny is going to be speaking to us about HLA eplet frequencies and genetic complexity for an eplet-matching program in kidney transplant. Great science. So come on up, Jenny. And each speaker there'll be a 2 to 5 minute question period after each of their presentations.
Hi, everyone. So yes, my name is Jenny Tran, and I'm a PhD student in the Department of Experimental Medicine at UBC. Thank you to the organizers for, is this okay? Okay. Thank you for the organizers for providing the opportunity for me to speak today. My presentation is titled HLA Eplet Frequencies, Reduce Genetic Complexity, and Provide a Foundation for a National Eplet-matching Program. There are 2 learning objectives. The first is to define the role of HLA eplet matching in kidney transplantation, and the second is to describe HLA allele and eplet frequencies in kidney patient and donor groups in BC. Kidney transplantation is the treatment of choice for those suffering from end-stage renal disease. It is associated with reduced risk of comorbidities and increased quality of life compared to dialysis. Unfortunately, the probability of kidney transplant failure increases days after transplantation, where many of these failures are due to antibody-mediated rejection, or AMR. AMR occurs when the recipient's immune system makes antibodies targeted against foreign proteins expressed on the donated kidney. These antibodies can mount a cascade of immune responses that will attack the donated kidney, leading to graft rejection. The proteins that these antibodies bind to are called the human leukocyte antigens, or HLA. HLA are heterodimeric proteins expressed on the cell surface. They're essential for the adaptive immune system to respond to foreign molecules. And there are 11 clinically relevant HLA genes on chromosome 6, divided between the class 1 and the class 2 genes. We know that antibodies don't bind to the whole HLA protein themselves, but rather to sections of amino acids on the surface of the protein. Eplets are clusters of 2 to 5 polymorphic amino acids that are essential to antibody binding. Eplet matching is matching patients and donors based on their HLA eplets. And it's based on the theory that the recipient's immune system will not mount a response against eplets shared between the patient and donor. So if you imagine an HLA protein as a string of immunogenic eplets, as shown in this cartoon diagram here, the patient HLA is composed of 4 eplets. And this patient could be matched against 3 potential donors. Donor A has 4 eplets, but none of these eplets are shared between the patient and donor. Thus, there are 4 eplet mismatches. However, if we scroll down, if we go to donor C, we can see that 3 out of the 4 eplets are shared between the patient and donor, and only the pink hexagon is not. So therefore, this is a 1 eplet mismatch, and donor C would be the best candidate for this patient. Multiple studies have shown that eplet matching results in improved transplant outcomes. Importantly, there are far fewer documented eplets than there are alleles. The aim of this study was to describe HLA allele and eplet frequencies in kidney patient and donor groups in BC and compare the likelihood of matching by each method. We've sequenced 1,846 patients and donors at their HLA genes. We then converted the resulting alleles into eplets using a computer algorithm, and then we performed our data analysis. One of our main findings was that the conversion of alleles to eplets greatly reduced HLA complexity. For example, for HLA-B gene, we've identified 107 eplets. 107 alleles. When we converted the alleles into eplets, we identified only 26. This was a 76% reduction in HLA complexity. When we map the relationship between alleles and eplets, we can understand that this reduction in complexity was due to the fact that multiple alleles encoded for the same set of eplets. For example, 131S eplet, shown in red, is actually encoded by 90 alleles. So although we identified 107 B alleles in our population that would translate to 107 proteins, on the eplet level, these proteins are actually more similar than we originally have thought. Overall, we've identified 361 alleles that converted into 150 eplets, resulting in a 59% reduction in genetic complexity. Class 1 alleles encoded 0 to 11 eplets each, and class 2 alleles encoded 0 to 17 eplets each. We then determined the relative frequencies of alleles and eplets in our population. So this was defined as the number of times an allele or eplet occurred in a group, divided by the total number of individuals in that group. And our total population was subdivided into 4 patient and donor groups: KTX for transplanted kidney patients, K-pre for pre-transplant patients, LD for potential living donors, and DD for deceased donors. And this is our results for our class 2 allele frequencies. It might be a little bit hard to see, but on the bottom x-axis are all our class 2 alleles that we've identified in our population, plotted against their relative frequencies from 0 to 1 in their respective patient and donor groups. And we can see that allele frequencies occur at very low values, where the majority of alleles occurred in less than 10% in their respective patient or donor group. However, when we perform the same analysis for eplets, we can see that there's a dramatic increase in relative frequencies, so that eplets are more common in the patients and donors than their allele counterparts, giving evidence that eplet matching may be more feasible than allele matching. Next, as a means to assess the similarities between patients and donors based on their eplet frequencies, we plotted their frequencies against one another in a pairwise analysis. For example, on the top right plot C, each dot represents an eplet. And on the x-axis is the relative frequency of those eplets for our transplanted patients. And on the y-axis is their frequencies for deceased donors. And we can see here that this relationship is a very close linear relationship with a high correlation coefficient, indicating that these eplet frequencies are similar between our patients and donors. And we actually saw this in all pairwise comparisons in our donor and patient groups. Lastly, we performed a clustering analysis to identify clusters in our total study sample based on eplet expression patterns called an HLA eptype. A cluster represents patients and donors whom express similar eptypes, and we were able to identify 3 clusters in class 1 and class 2 genes. For example, this is a heat map depicting the clustering analysis for class 2. So the first row for C1 represents cluster 1, and it represents patients and donors who express similar eplet expression profiles, and is driven by high expression of the eplets boxed in red. Individuals in cluster 2 and 3 do not express these eplets at very high frequencies. The same can be said for cluster 2, where patients and donors that cluster into cluster 2 express these eplets at high frequencies, and those in cluster 3 do not. This informs the type of matching we could expect to get if we were to perform matching based on eplets, where, for example, donors that cluster into cluster 1 will most likely donate to patients in cluster 1 because they share similar eplet expression profiles. In conclusion, eplets reduce the HLA complexity in a BC study sample. Patients and donors show similar eplet frequencies, and this study sample grouped into 3 clusters expressing similar eplet profiles, of which we've called eptypes. Overall, the results support the feasibility of eplet matching in BC and efforts to improve long-term transplant outcomes. So thank you. I would like to thank the organizers again for allowing me to speak today, as well as the granting agencies and the immunology lab at VGH and the help from the Genome Canada Transplant Consortium. Thank you.
I'm going to start off with the first question, which I think is a gift of a question to you. There's a lot of people in the audience, myself included, who aren't immunologists and who aren't really even transplanters. So I wonder if you could, maybe we'll just take a huge step back and explain for us maybe what the significance of eplet matching would be. What would this mean to a patient? How would a patient benefit from this?
Yeah, definitely. So eplet matching is a means to improve the way we allocate our donors to our patients, so they can improve in the long term. They can have their kidney for a longer amount of time, so that they don't have to go back for retransplantation or dialysis. And as well, if the donor is matched to the patient, like for example, for identical twin transplants, immunosuppression could be reduced. And so if we can try to better match our patients and donors, we may lead to better clinical outcomes for the patient.
That sounds great. That sounds like what matters most for a transplant recipient, so that's good. No one's posed any other questions. Any questions from the microphone? I have one other here. What do you think would be the next steps if we were to move forward provincially with eplet matching, especially to ensure equity in organ allocation?
Yeah, so a lot of work still needs to be done in this field. One of the things that my lab is trying to do is simulate eplet matching in the same cohort that we perform this analysis in. So this is kind of like a test run before we actually do it. So what we would do is we'll have a simulated donor pool and then a waitlist pool. And we would simulate one random donor picked from the population and match it against the waitlisted patients, and then we will analyze to see what type of matches we get, and we'll repeat this analysis to see patients who may be disadvantaged by performing eplet matching, the types of matchings, the type of mismatches we would get, and yes, to better inform eplet matching if we were to use it in a clinical setting. Thank you.
Super. Thanks very much, Jenny. Next, I'm going to call on Amanda Driver. Amanda's a clinical pharmacist at Fraser Health, and she's going to speak to us about an evaluation of the Empiric Antibiotic Regimen for treatment of PD-associated peritonitis at VGH.
Hi, everyone. I'd also like to thank the organizers for the opportunity to present today. So my presentation is titled An Evaluation of the Empiric Treatment of Peritonitis in Peritoneal Dialysis Patients at Vancouver General Hospital. So peritonitis is one of the more serious complications that can occur in patients undergoing peritoneal dialysis. It often leads to unfavorable outcomes such as hospitalizations. Due to the structural and functional changes to the membrane during infection, it can also lead to peritoneal membrane failure. Patients may have to transfer to hemodialysis either temporarily or permanently, and it also leads to an increased risk of mortality. So the International Society of Peritoneal Dialysis published new recommendations for peritonitis treatment in 2016. This was an update to their previous recommendations from 2010. So they suggest that at the first suspicion of peritonitis, after you've obtained a dialysate sample, that you initiate empiric intraperitoneal antibiotics plus antifungal prophylaxis. So this was a bit of a change from the 2010 guidelines. They previously recommended antifungal prophylaxis at the discretion of the nephrologist based on the patient-specific risk factors for fungal peritonitis, but they're now suggesting, based on some new literature, that perhaps all patients receiving antibiotics have this antifungal prophylaxis. So when we treat with empiric intraperitoneal antibiotics, they recommend covering for both gram-positive and gram-negative organisms. To cover the gram-positives, they suggest a first-generation cephalosporin or vancomycin, and to cover the gram-negatives, they recommend either a third-generation cephalosporin or aminoglycoside. For antifungal prophylaxis, they recommend oral therapy with either fluconazole or nystatin. They also mention in the guidelines that we don't really have any good evidence of a superior regimen for the treatment of peritonitis, and that it should be based on each center's specific rates of resistance and what they found works best in their population. So based on this, we looked at our current practice at VGH, and that's for empiric intraperitoneal antibiotics. We provide cefazolin, which is a first-generation cephalosporin, to cover the gram-positive organisms, and then ceftazidime, which is a third-generation cephalosporin, to cover the gram-negative organisms. Exceptions would include if the patient has a cephalosporin allergy, or if they have a history of resistant infection, specifically MRSA or MRSE, that we would provide vancomycin and tobramycin. And then for our antifungal prophylaxis, we do use fluconazole, and it's prescribed based on patient-specific risk factors for fungal peritonitis. The objectives of our project were to first characterize the pathogens and the resistance patterns of the peritonitis episodes over the past 5 years. We wanted to evaluate the effectiveness of our empiric antibiotic regimen based on the clinical outcomes. And then we wanted to assess our fungal peritonitis rates over the past 5 years to determine whether we needed to adopt this new practice of prescribing antifungal prophylaxis to all patients. So we conducted a retrospective chart review of PD-associated peritonitis episodes at VGH from January 2013 to December 2017. These episodes were identified using the PROMIS database. We included adult patients who meet the criteria for PD-related peritonitis. So the criteria was based on the ISPD guidelines. Patients must meet 2 of 3 criteria. So the first one being they have clinical signs or symptoms of peritonitis, which is usually abdominal pain or cloudy dialysate. Number 2, that their dialysate analysis shows a white blood cell count greater than 100 per microliter, with greater than 50% of those white blood cells being neutrophils or polymorphonuclear cells. And then the third criteria is if they have a positive dialysate culture. We only included patients who did receive empiric treatment of their peritonitis. And then for exclusion, we excluded any patients who were found to just have an exit site infection, or if they had eosinophilic peritonitis. So also from the PROMIS database, we obtained the peritonitis rate. So this is defined as the number of episodes per patient year on PD. The rate here is for the province as a whole and then VGH specifically. So over time, the rate has been declining. So for VGH in 2013, the rate was 0.33, and by 2017, it's 0.09. The ISPD suggests that an acceptable rate is anything below 0.5. So when we looked at the patients identified, we found 42 patients who had peritonitis episodes over the 5-year period. 18 of these were male. The mean age was 65, and in these 42 patients, there were 62 episodes of peritonitis. So there were patients who had repeat episodes. For dialysis modality, 75% were on continuous cycling PD and 25% on continuous ambulatory PD. And then the mean duration of dialysis was 938 days. However, you'll notice there's a large standard deviation, so we weren't really able to correlate the incidence of peritonitis with how long patients have been on peritoneal dialysis. When we looked at some of the risk factors for exit site antibiotic to prevent infection, about 50% of our patients were using mupirocin and 50% were using gentamicin. Prior to their peritonitis episode, only 1 patient was known to be MRSA positive. 8 patients were on immunosuppressive therapy. Usually those patients were previous transplant patients. 15 patients had received antibiotics in the past 3 months, which is a known risk factor for fungal peritonitis. And then 3 patients had extraperitoneal fungal infections. 2 of these were exit site infections. So when we looked at the empiric therapy that was provided in the 62 episodes, to cover the gram-positive organisms, 77% of our patients received intraperitoneal cefazolin, with 16% receiving vancomycin. The other category composes patients who perhaps lost access and received some other type of empiric therapy, either IV or some other type of antibiotic. To cover gram-negative organisms, 82% received ceftazidime and 7% tobramycin, and again, 11% in the other category. And then 25% of episodes received fluconazole for antifungal prophylaxis. So when we looked at the organisms that we isolated, in the 62 episodes, there were 74 organisms. So some had polymicrobial infections. The majority of the organisms were gram-positive, so Staphylococci making up 39%, Streptococci making up about 18%, and then gram-negatives made up about 30% of the isolated organisms. We found we had no episodes of fungal peritonitis over the 5 years, and our culture-negative peritonitis rate is 9.7%, which for reference, they suggest an acceptable rate is anywhere between 10 and 20%. We then looked at the antibiotic susceptibilities of the organisms we isolated. So for our Streptococci species, there were 13 isolates, of which 77% were sensitive to penicillin, from which we infer that they would be sensitive to cefazolin. 100% were sensitive to vancomycin. For Enterococci, 83% were sensitive to vancomycin, and then gentamicin, 67%, which it could be used for synergy. For Staphylococci, there were 29 isolates. 92% were sensitive to cefazolin, and then of those tested, 100% were sensitive to vancomycin. For gram-negative isolates, 100% were sensitive to ceftazidime. If they weren't sensitive to ceftazidime, they were sensitive to ceftriaxone. This graph shows our peritonitis episode outcome, so I'll just orient you to the figure. So on the y-axis is the episode outcome. On the x-axis is the number of episodes that achieved that outcome. In light purple is episodes in which the organism that we isolated was sensitive to the initial empiric therapy provided. In dark purple is episodes in which the organism we isolated was found to be resistant to the empiric therapy that the patient received. So in 55 episodes, the organism was sensitive. We achieved resolution of infection in 50 episodes out of 62. The most common other outcome was refractory infection, and so that's defined as failure of the dialysate to clear after 5 days of antibiotics, and that occurred in 7 episodes. We had 1 death associated with peritonitis, and then 13 patients had their peritoneal dialysis tube removed. They were all transferred to hemodialysis, and 3 of these patients were able to transfer back to peritoneal dialysis. We identified a few limitations and confounders that may affect our data. So we did have a small sample size, and we're unable to associate any patient characteristics with clinical outcomes. Again, we used the PROMIS database to identify our peritonitis episodes, which is dependent on us reporting our peritonitis to PROMIS, so that may have underestimated the sample. Patients with multiple peritonitis episodes may impact resistance patterns. We did look at this, and I believe 50 of our 62 episodes, the patients were on either their first or second episode of peritonitis. And then we found additional antibiotics were administered in about 1/3 of episodes. And this was most commonly like a single dose of antibiotics in the emergency room before they received their intraperitoneal antibiotics. So that could also affect the outcomes that we saw. So in conclusion, over the 5-year period, 89% of our episodes were sensitive to the empiric therapy that we were providing. Therefore, we concluded that our current empiric antibiotic regimen is adequate for the treatment of PD-related peritonitis at VGH. And then because we had no episodes of fungal peritonitis over the 5-year period, we have suggested that routine antifungal prophylaxis is likely not indicated, but that continuing our current practice of prescribing antifungal prophylaxis based on risk factors would be appropriate. Thank you, and I'm happy to answer any questions.
Great. Yeah.
Hi.
Michelle?
I'm just curious, with a different empiric regime, if you calculated what the success rate would have been. Say you'd use something like cefazolin and an aminoglycoside, or aminoglycoside and vancomycin, if you actually calculated out those different regimes and what your success rate would have been.
We did not perform that analysis, but that's a very valid point. The results we presented are like some patients did receive vancomycin, but then if the organism was susceptible, they were considered to be susceptible to empiric therapy. So it would be interesting to look at if we had provided different therapy, if our success rates would have been better.
It sounds to me like the VGH program is to be commended on the really low peritonitis rates. I know that you're their pharmacist, but I wondered if you might be able to offer any insights. The rates were lower than the provincial average. Any thoughts on that?
Yeah, we've discussed this a little bit as to there's quite a few known factors that can help to mitigate peritonitis. There's a lot of look into when we're inserting PD catheters, like the antibiotics that we provide, the technique for insertion. There's been lots of research into the connection systems that we used, but one of the most prominent things we identified is that there was a change a while ago in the nurses that work in the PD unit, and having specific dedicated nurses who are trained in the curriculum to teach patients how to perform peritoneal dialysis, and we think that that might also be a really important factor in lowering the peritonitis rates.
I was interested in your comment and the conclusion, and I can see why you came to that conclusion about not needing fungal prophylaxis, but we're really vigilant anytime any of our PD patients over at Island Health gets put on an antibiotic, they get put on mycostatin. So it's interesting to look at ISPD and see that you could give mycostatin or you could give fluconazole. To me, the fluconazole is this pretty big gun. Lots of interaction problems, and that's why we're not using it. But, oral swish and swallow mycostatin seems comparatively feeble, shall we say, and I wonder if it's doing anything at all. Do you have any thoughts about that, or is there any data on its effectiveness, if any?
There's a bit of data. I agree. I think the main barrier to the nystatin is the 3 times a day dosing and the swish and swallow. But then, as you mentioned, fluconazole has lots of drug interactions and is not benign. So I think there's limited data there is data on that antibiotics do increase the risk of fungal peritonitis, and they found that most fungal peritonitis episodes occur after exposure to antibiotics. So I think there's good data there. As for the data for the prophylaxis portion, I think we have limited studies on that. But again, the frequency of fungal peritonitis in your specific population would also help to guide therapy.
One final question was, I recognize your data is for VGH, and you may not know provincial data, but would you foresee the, perhaps not the types of organisms, but the regimens, treatment regimens, or peritonitis in any way being different anywhere else than at VGH?
Yeah, I think the ISPD is very valid in that each program is different, and it's based on your patient population. And I know that there are programs throughout the Lower Mainland that use different empiric therapies. Some people use vancomycin up front, and that's based on your population and what the most frequent organisms are and the rates of MRSA. I think really this data only applies to our specific population, but I think similar initiatives could be taken in other areas to look and see what the empiric treatment should be.
Thanks very much.
Thank you.
Okay, so we've got, again, moving on with a nice range of topics. Our third oral presentation is to be presented by Leonora Chow, and she's going to speak to us on the effects of a renal Nordic walking program on quality of life and fitness in renal patients at St. Paul's Hospital. So welcome.
Good morning. We're all honored to be here today. Thank you to the organizers for selecting our research to share. I'm Leonora Chow, team leader and PI for our research study titled Effects of Renal Nordic Walking Program on Quality of Life and Fitness in Renal Patients at St. Paul's Hospital. I've invited my co-investigators on stage with me to assist with the Q&A session that follows. From left to right is Vicky No, Randy Renal, and Susie Neufeld. Susie is the only physio on our team. The rest of us are dietitians. Our study was funded through the PHC Practice-Based Research Challenge competition. We know that a healthy diet and exercise is important for good health, but many of our renal patients don't meet the activity guidelines. So a group of staff volunteers created a Renal Nordic walking program titled the Walk and Row in 2016 to support our patients in becoming more physically active. And this was done knowing that hospital-affiliated, group-based, renal-specific exercise programs that provide opportunities for peer support and supervision can potentially reduce fears and concerns and increase exercise self-efficacy. This is a picture of the Nordic walking pose that we use for our study, and Danny has a pair with her. We'll have them during our e-poster presentation later on this afternoon at 2:30, so feel free to drop by and you can try them out. You can see the bottom of the pose is a bell-shaped rubber tip, which provides stability, and the handles are ergonomic and they don't have straps. Straps can cause injuries if a fall was to occur. So although walking is usually the best accepted form of exercise for general health, Nordic walking can boost confidence, help with balance, particularly in older adults. Other studies have shown that Nordic walking to be superior to brisk walking without poles, such as more calories burned without an increase in perceived exertion, a commitment to an exercise program, and improving upper body strength. We hope that by sharing our pilot study results with you today, it will support the growth of the St. Paul's Hospital Walk and Row program by increasing patient participation and exercise self-efficacy. Also foster the promotion of physical activity as part of renal patients' clinical care in improving fitness and quality of life, and lead to future research or other initiatives. This is a picture of the Nordic walking toolkits that all participants in our study received. There's a t-shirt that's designed by our patient partner, a Fitbit tracker, a walking fitness handbook, and a nice cloth bag to carry your supplies during the walk, like a glucometer, snacks, and water. And also, the pair of Nordic walking poles were provided or given to our patients after the 12-week study period. Our study was a randomized control trial where we recruited 30 participants and randomized them to the Nordic walking group or the control group. So the Nordic walking group, they were offered 2 walking sessions per week at St. Paul's Hospital that were supervised. We kept track of attendance. The control group, they continued with their own activities for the 12-week study period. All participants were provided with a Fitbit tracker. They kept track of their daily steps for the 12-week study period. The inclusion criteria included anyone from the age of 19. They had to be a renal patient. They could be CKD, PD, hemo, or transplant. They needed physician's approval, were able to walk safely, and they had a smartphone and were comfortable using the apps. The data that we collected at baseline at the end of the 12 weeks included quality of life questionnaire, hand grip strength, sit to stand test, the 6-minute walk test, and weight. Unfortunately, no blinding of intervention or outcome assessment was possible, meaning that we knew which arm the participants were in when we collected the data at baseline at the end of the study. Research methodologists from KIOS assisted us with the data analysis, and median changes in outcomes from baseline to 3 months were calculated between groups. You can see in this slide that we had equal numbers in each arm of the study, control and intervention. The median age of participants was about 66 years. We had equal numbers of males and females. 14 of our participants were CKD, 3 were on hemodialysis, 3 peritoneal dialysis, and 10 from transplants. In this slide, it shows that our randomization was not equal, perhaps due to the small sample size. Highlighted in purple is the control group. The highlighted in yellow is the Nordic walking group. From this slide, it appears that the Nordic walking group was less healthy than the control group. The Nordic walking group had a higher BMI, 28 versus 24. They had a lower 6-minute walk distance, and they also had lower quality of life scores. These box plots show the changes in 6-minute walk test and hand grip strength from baseline to the end of the 12-week study. The red box is the Nordic walking group, the blue box is the control group. Interestingly, the Nordic walking group showed clinically meaningful improvements in the 6-minute walk test at the end of the study period. The Nordic walking group had a 41.5-meter increase in 6-minute walk test with a confidence interval of -3 to 89 meters, which skews positively up. In contrast, the control group only had a 10-meter increase in 6-minute walk test, which according to the literature is not clinically meaningful. A systematic review in 2017 reported that a 14 to 30-meter improvement might be clinically meaningful across a multiple of patient groups. In renal disease, a study showed that a 6-minute walk test that was less than 350 meters was associated with a 2.8-fold increased risk in mortality, and that for every 20-meter improvement, reduced mortality risk by 11%. In the right box plot, you can see that although not statistically significant, the Nordic walking group also had greater improvements in the hand grip strength. This slide shows the changes in quality of life from baseline. Again, the Nordic walking group is shown in red, the control group in blue. You can see that there are significant improvements in several domains of the quality of life questionnaire: effects of kidney disease, burden of kidney disease, symptoms and problems of kidney disease. I think that the comments from the participants, you can see some of the comments at the bottom of the slide. These comments were after the 12-week study period. I think these comments resonate the theme of this year's BC Kidney Day, what matters most. And these comments also confirms an improved quality of life. So basically, some of our participants found they were sleeping better, they had more energy, it took less effort to do things, more confidence in exercising. Missingness analysis showed that 93% of the data is present. This is quite high. And so this strengthens the confidence in our finding. Only 2 participants dropped out from the study for medical reasons unrelated to the study. It was unexpected and quite remarkable that the Nordic walking group appeared to have better results despite appearing less healthy at baseline. And this bodes well for a walk and roll program, as the most frail are probably the most in need of physical rehabilitation as part of their clinical care. The Nordic walking group tended to have better outcomes despite appearing to have a lower daily step compared to the control group. And we should interpret these daily steps with caution because many of our patients had problems using their Fitbit. There appears to be benefits in the walk and roll program, which we weren't able to discern because there was no correlation between attendance and better outcomes. The median attendance was 14 days out of the 23 sessions. We continue to disseminate our research findings in a variety of ways. This is a screenshot of the daily scan that came out in May, and it was just reposted again yesterday on Facebook. We're happy to report that the PHC Renal leadership has allocated funding for a physiotherapist to run the walk and roll next year. We're also exploring partnership with others in the community. We're inviting UBC students to volunteer to get involved in rehab. We'd like to acknowledge our partners in our study, without whom their support and contribution, none of this would have been possible. I'd like to open up the floor for questions.
Thank you. Super interesting and nice to hear this theme also about the active patients today. There's a couple questions coming in. The first was if this program was going to be offered to community dialysis locations.
Currently, it's just going to be at St. Paul's Hospital.
Sounds like we can all read your results and share the excitement and maybe bring that back to our own program. So That information will be posted at BC Kidney Day at our website as well, if anybody needs to look into that. I'm curious if you shared the results with other patients. Was this broadcast in the dialysis unit, in the kidney care clinic? To me, it would be of tremendous benefit for other patients to see the real results with what you're saying, pretty modest input from others.
Absolutely. So we have plans to share the information with the Chinese Renal Association, because they were the first participants of our Renal Nordic walking program. We're also brainstorming about ways and sharing this information with our patients, perhaps in the clinic setting.
What are some of the strategies that you use to encourage participation and attendance, apart from the lovely gifts?
We didn't really have to encourage attendance. I think it was astounding to us that simply walking around the perimeter of Nelson Park became so important to this group. They came from Richmond, Coquitlam, twice a week with, dreadful parking around St. Paul's, but we did try to help out there. But they also, they established a social bond between one another. And they also indicated to us over and over again how much they appreciated the fact that we as professionals were taking time out of our day and were doing this simple walk. During the walk, they shared their stories. They asked questions about nutrition, tennis elbow, heel spurs, It went from beyond just walking to be more comprehensive, and I think that speaks to the key issue here of humanity.
I think it's also important to mention the capacity that a program like this can build above and beyond what we might expect as researchers looking at these quality indicators. But I think our volunteers and their engagement in this program has been instrumental as well in building capacity for young students who want to train to be professionals, whether physiotherapists or dietitians or other types of healthcare that they're exploring. So I thought that was an important piece. The volunteers were really engaged and involved as well. So it just, leverages what we're doing so much more when we have people from different areas in the community coming together like this.
Seems like a good opportunity to get the UBC students involved. You're mentioning as well some.
Yeah, absolutely. So I started off with 3 volunteers, and I now have 12.
Okay.
And they all want to do the Nordic walking. So
nice?
yeah, it's great.
Last question then will be, how much do the poles cost and where can they be purchased?
Okay. The polls were from Urban Polling. They have an online website, and they're about $100. But you can get them at other, like Shoppers Drug Mart, and sometimes they have sales.
Thank you very much.